And dear students of 12th class, and today in your botany we are going to take up the Mendel's law of independent assortment. For this, we have to take a look on the previously studied or discussed the dihybrid cross. In the dihybrid cross, Mendel made the cross between the two parents which differed in two pairs of contrasting traits. Here you can see the two pairs of the contrasting traits were the seed color and seed shape. Here you can visualize this is the parental generation wherein we have a round and yellow seed with green. The cross was made between the plants with the round and yellow seeds with the plant that was having wrinkled and green colored seeds. Obviously, the round shape was dominant over the wrinkled shape of the seed, and the yellow color was dominant over the green colored seed. And in the gametes we have only the one out of the two allelic pair. We get only the one factor that is transmitted to the gametes, and here it is represented by capital or small y for the round yellow, and the gametes are represented in case of the wrinkled green by small r and small y, indicating the wrinkled seed shape and the green color of the seed. And in the F1 generation, as you may be knowing, that only the dominant character is expressed. All the seeds produced are round and yellow, which are then allowed for the selfing, and the seeds thus obtained are in the ratio of, in case of a dihybrid cross, in the ratio of 9:3:3:1. 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. And this was also true for the other pairs of contrasting characters when studied together in a dihybrid cross by the Mendel. The ratio was again the same, that is 9:3:3:1 in the F2 generation. And now we will study the law of independent assortment, that is also based on the dihybrid cross. So as we have already seen in the dihybrid cross, the phenotypes round yellow or wrinkled yellow, round green, and a wrinkled green appeared in the ratio of 9:3:1. Such a ratio was observed for several pairs of the characters that Mendel studied. As I have already told you, the ratio of 9:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round, 1 wrinkled, and this derivation can be written as follows: 3 round is to 1 wrinkled, 3 yellow is to 1 green, and 9 round is 9 round yellow is to 3 wrinkled yellow, and 3 round green is to 1 wrinkled green. And that is the ultimate ratio of 9:3:3:1. Now, based upon such observations on dihybrid crosses, as the crosses between the plants differing in two traits, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. And this law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. That means the characters The genes carrying the characters, they do not intermingle, and they carry the characters independent of the other genes carrying the different characters. Again, this law of independent assortment states that when two pairs of traits are combined in a hybrid, segregation of one pair of the characters is independent of the other pair of the characters. They do not influence one another. The Punnett scale can be effectively used to understand the independent segregation of the two pairs of the genes during meiosis and the production of the eggs and the pollen in the F1, that is capital R small r, capital Y small y plant, the hybrid plant with the dominant phenotypic characters. Consider the segregation of one pair of genes, that is capital R and small r, for a round and wrinkled shape of the seeds. 50% of the gametes have the gene R, and the 50% have 50% with capital R and 50% with small r. Now, besides each gamete having either capital R or small r, it should also have the allele capital Y or small y, as we are studying two pairs of the characters together. The important thing to remember here is that the segregation of the 50% R and the 50% R, small r, is independent from the segregation of the 50% of the capital Y and 50% of small y, that is for the other characters. Therefore, 50% of the small r bearing gametes and the capital Y and the other 50% of has small y. Similarly, 50% of the capital R bearing gametes has Y, capital Y, and the other 50% has small y. Thus, there are 4 genotypes of gametes, 4 types of the pollen, and 4 types of the eggs. The 4 types are represented by, as you can see in the Punnett square, like this: capital R, capital Y, small r, capital Y, capital R, small y, and small r, small y. And now, each with a frequency of 20% or 1/4 of the total gametes produced. When you write the 4 types of the eggs and the pollen on the two sides of the Punnett scale, it is very easy to derive the composition of the zygotes that give rise to the F2 generation plants. Although there are 16 squares, now how many different genotypes and phenotypes are formed, that can easily be visualized in the Punnett square. Here, when we study the two pairs of the characters, as you can visualize that there are seeds. If we count the yellow round, there are 9 in number. If we count green round, there are 3. If we count yellow wrinkled, also 3 in number, and only 1 that is wrinkled green. And the ratio here is 9:3:3:1 for a dihybrid cross. That means when we study two pairs of the contrasting characters together. And this was the law of independent assortment. Hope you will study it from your notes and the study material that you have also, and you will be able to grasp it easily. Thanks.